Well, welcome everybody. My name is Andrea. I am the Adult Programs Manager at the Fleet Science Center, and I'm super excited that you are joining us again for another Suds Science talk in our special series, San Diego's Race to Combat COVID-19. I'm very excited about this series as it gives us a chance to highlight all the amazing science that is happening in San Diego. Although it's a very sad case of why we get to showcase San Diego and all the work that's being done. San Diego is an amazing science city. I'm awfully proud of that fact, and I hope that many more people will join me in recognizing and being proud of San Diego being such a great science city. Today we have Dr. Erica Ollmann Saphire with us from the La Jolla Institute of Immunology. And before we get into the talk, just a couple of housekeeping things. Your camera and audio is turned off, but we would love to hear your questions, and we have some Q&A time set aside at the end. There is a Q&A function at the bottom of your screen. If you want to click that, you can type in your question at any time. We will get to them after the presentation from Dr. Ollmann Saphire. But type them as soon as it comes to your mind, type it in there, then we're ready to go when we get to the Q&A function. And I think with that, I am going to turn it over to Dr. Ollmann Saphire.
Hello, it's nice to see you, and thank you everyone for joining tonight. So I'll start by sharing my screen. I am a professor at La Jolla Institute for Immunology. So I'll show you a picture of what the place looks like. We are a private biomedical research institute on the UCSD campus. We are faculty or adjunct members of the UCSD Department of Medicine, but we're private and independent. And that means that we focus entirely on research and training of graduate students and postdoctoral fellows. And we focus solely on immunology and the immune system and what we can learn from different ways of studying the immune system to help us fight different diseases. I am a virologist, and so as you can imagine, my lab has been turned over to coronavirus research. So tonight I'd like to tell you a little bit about where we are with this virus, immune defense, and then a global consortium that I'm running from La Jolla. So where we are right now is that we've had 2.1 million cumulative cases of coronavirus in the United States and about 120,000 deaths. And you can see there is a fairly level number of cases. It's not gone away, and in many places it is increasing. So you can see when we were given that shelter-in-place order in mid-March, there's a relatively small number of cases then. In order for someone to identify as a case, they're typically about 2 weeks after that day when they got infected. So 2 weeks later, you can already see that large ramp of cases. Those are the people that got infected before or right about when that shelter-in-place order was given. Then it started to decline, but it's holding steady, and we're seeing in some places that it's getting worse. So in some states, it seems to be declining, and these are a lot of states where they had an early burden of infection. In other places, new cases are increasing, some with steep trajectories, others more steady. Now, because I'm a scientist, I like to put things on the same scale. So if you put California in the same scale as New York, you can see New York really had a significant problem in early April. We haven't gotten there yet, and I'm hoping we never do. But that was the scale of that problem from 4 different relatively populous states. Now, to give you a couple ideas of some markers in time, this is when our shelter-in-place order was given. This is about 2 weeks after that. Here's Easter, 2 weeks later. Here's Memorial Day, 2 weeks later. So we're seeing an increase in cases. Certainly we have an increase in testing, but I think there's a real increase in cases. And where we are right now is there. So what's going to happen next? Well, we can probably expect the obvious thing, which is as we go out more and interact with people more, there will be more cases. We do see that the most common cases now tend to be among younger people who are venturing out. And they typically fare quite well, not always, but much better. And it looks like we're going to have this virus with us for a long time. We typically are more susceptible to respiratory infections in the fall and winter. Now, these novel ones that have just spilled over, they don't always follow the normal patterns. But we are expecting cases to continue rising through the summer, and it could get much worse in the fall and winter, or it could be a recurrent seasonal infection if we haven't fully eradicated it from humanity. So what do epidemiologists think? Well, the New York Times did a survey of 500 epidemiologists and said, well, what would you do in your private life? When would you be willing to do these things? And here are some things they would, not all, but half maybe would be willing to do this summer. Go to a doctor for a non-urgent appointment, vacation overnight, get a haircut. It's interesting that more of them are willing to go on vacation out of town than to get a haircut. Maybe that means that epidemiologists right about now need a vacation, or maybe they've learned how to do home haircuts. But even fewer would do these things right away, and it might not be until winter or next spring before they're willing to go to a dinner party or ride a subway or a bus or travel by airplane. And then things they won't do for another year, this suggests that we might reach a new social normal. They would be a bit resistant to hug or shake hands, ways in which we normally greet each other, go to church or religious service, or stop wearing a mask. So we're going to have to get used to this new normal until we have immunity. And there is an easy way and a hard way to get immunity. The hard way is to get infected and get better. The easy way is through a vaccine, were there one available yet. There is hope that some will be available in 6 to 12 months, but we don't know, and we need to see that data, and they need to be tested. But in order to have enough herd immunity that you can rely on the people around you to be immune, to be that fire break, we think it's going to require about 60 or 80% of the population to be infected and have gotten better. So the minimum 60% herd immunity looks like the left, where an immune person would be a purple dot, non-immune person would be a gray dot. We're not there yet. Even New York City, that has significant problem, is only about 20% seroprevalence. Only 20% people have antibodies. Stockholm, only 7%. And we have had a lot of deaths to get to that point. In the US overall, not in these densely populated cities, we're probably at just a few percent herd immunity, but 120,000 people have died to get us to that point. And it's not necessarily nothing for the people to get better, because it can take them weeks and months to get better, and there's some long-term health consequences. So what we really need are vaccines and treatments to get us there and get us immune. Now, the very earliest optimistic estimates of when we might have vaccines are about January, which is a few months from now. Other estimates say it won't be until late summer or fall. Either way, it looks like it's going to be about 6 months to a year before we can rely on vaccines. So we need something to get us through to that time point. The other issue is that the important date is not the day the first person can go to the first CVS and get a shot, but the day that the globe is broadly protected. Because it's been very clear from this outbreak that a few infections in one place in the world could rapidly spread absolutely everywhere. And if there are pockets of the world that are not protected and who are not vaccinated and who are not immune, that virus is going to come back. The other issue is that it's going to be a long time until we have been vaccinated. Until we get to that point, there are a lot of people, all of us, won't have been vaccinated. Even once vaccines are available, there will be a lot of people who haven't been vaccinated yet. There are others who can't be vaccinated. Typically, all babies under age 1, often pregnant women, people with underlying health complications like compromised immune systems, can't receive some kinds of vaccines. And so they are vulnerable. And other people, some elderly people, the vaccine doesn't take. And so you need a treatment or a way to deliver immunity for these people that is complementary or independent. And so a good way, it's not coronavirus, I inhaled some water. If you haven't been vaccinated, or you haven't been vaccinated yet, or you're one of the groups that can't be vaccinated, an option could be an antibody therapy. So antibodies are molecules that your own immune system makes when you have gotten better from an infection or when you have received a vaccine. You can take a couple a couple of days, couple of weeks, sometimes months to get the really good antibodies. So what happens is your immune system makes this response of these Y-shaped protein molecules. They'll circulate in your blood, and their job is to be incredibly precise. They have fingerprint-like recognition. They'll seek out and anchor on to a pathogen when they see it again and recruit immune system help to destroy that pathogen. They are remarkably precise. An antibody against coronavirus will only attack coronavirus. An antibody against chickenpox will only attack chickenpox. You can also get them through vaccination, but we don't have those vaccines yet. Right now, the antibodies we could look at are those from the people that have already survived. So an antibody is shaped like this. This is the IgG type of antibody. It's shaped like a Y. It's got two arms that point up and a base that points down. At the end of the arms that point up are combinatorial fingers. They're loops that are hypervariable. And this is what gives that antibody fingerprint-like recognition for a particular place on a particular pathogen. It fits together like a lock and a key, and the antibody has evolved to be just precisely attacking that one place and that one pathogen. The base, the bottom of that Y, is more constant in sequence and structure, and that is the part that signals to the immune system, I am attached to an invader, come and destroy it. And that is how you can achieve very precise. I am going to have to close this Q&A right now because I can't see my own slides. You'll have to show it to me later. Okay, that is what causes the antibodies to be remarkably precise. Absolutely perfect shape recognition for one particular pathogen. So that's you, those are pieces of the virus. And the wonderful thing about antibodies is that they are so effective at anchoring to and inactivating a virus that you can deliver them like a drug. So you're familiar with antivenom. Those are antibodies against snake toxins or spider toxins. And you know that if you get a snake bite, you can get the antivenom, and right away you have immediately antibodies that will find and neutralize that toxin. And you know with antivenom that earlier is better. The same thing with antiviral antibody. You know after a rabies bite, you can get gamma globulin. That's antibody as well. And there are a number of diseases for which you can get other kinds of antibody treatments. The question is which antibodies to give. Because they have such a tremendous array of the sequences evolving to make so many different kinds of antibodies, there are a quintillion possible antibodies in human serum. That you find of the ones that are targeted against the pathogen you want to hit, the very best 1, 2, or 3 can be given as a drug. So in that tremendous array of antibodies you make, a lot are against something irrelevant. You know, it's good. They're against measles, they're against chickenpox, they're against polio. But if you want to find one for a drug against coronavirus, you need the maybe 1% that you're making against coronavirus, or a fraction of a percent. And of those, you want the very most potent few, not the ones that are duds. You want the ones that are most active, that anchor most tightly and work better. We have been able to find antibodies that are remarkably effective against all kinds of infectious diseases. There are some that you can put into premature infants to protect them from respiratory syncytial virus every winter. We have antibodies that treat autoimmune disease. We have antibodies that are used to treat cancer, to seek out and destroy individual cancer cells on a microscopic level in a way that you can never find with radiology or remove with a scalpel. It's a remarkably precise, beautiful therapy. And what's really nice about antibodies is that they are a natural human molecule. It's what your own immune system would make had it time and had it exposure to that antigen to do it. It's also transient. A natural antibody, if transfused into you, will last about 3 weeks. You can engineer it to make it longer lasting, maybe 3 months. It's not going to fundamentally change anything about your immune system, but it could get you through a crisis. And it's a typically very safe, often de-risked kind of therapy. And so it's pretty attractive because you can come up with an antibody therapy typically faster than you can a vaccine. It is often a very safe therapy because it's a natural human molecule. It's very precise if you can find ones that just exactly target the pathogen you want. And it makes a lot of sense to get us until that time point when we have vaccines and to treat those people that haven't yet been vaccinated or can't be vaccinated. So the issue is which antibodies do you deliver as a therapy? There are 50 or 70 companies racing right now to try to find the very best antibodies. And each one of them is using a slightly different strategy. They may have used a different representation of the virus. We call that an antigen, as slightly different bait. And you know, if you use different bait, you catch different fish. They may be using slightly different experiments to evaluate the antibodies they've gotten, so they're choosing them in different ways. And every one of them is a little bit different. And every one of these companies that are leading the pack and heading toward these clinical trials has also engineered a slightly different version of that base of the Y, the part that signals to the immune system. Some are aiming for a long half-life molecule. Some are aiming for something that they are assuming will not interact with certain immune receptors to make it, to remove the possibility that antibody can enhance uptake. So they're each engineering their molecules in different ways. And we need to know what the differences in these molecules are, which ones are best. We may not be able to test all of them because the number of beds available for clinical trials, the number of dollars available to spend in clinical trials might not be infinite. And you need enough patients at enough different stages of disease to get statistical significance. We also want to make sure that whatever it is we're testing in human patients is very good and very safe. So we need a way of figuring out which among these hundreds of therapeutic candidates that are advancing toward clinic are the best and what makes them best. The other issue is that we don't usually use these antibodies. Well, sometimes we do. You could use them by themselves or you can use them in combination. For viruses, especially RNA viruses that mutate, we like to use them in combinations. We call those combinations cocktails, because the virus can and will mutate and escape anything you throw at it. But if you throw not just one antibody, but a complementary pair of antibodies that hit the virus from different sites, it has a much harder time escaping from two things at once. And so often treatment of a virus with an antibody cocktail could give you more durable therapy. And so we're interested in finding out which antibodies you might, that not only are the best by themselves, but which ones you would mix together in the right combination. And it might be that the best combination is two molecules from discovered in two different labs. And you might not ever know that unless you had some way of putting them together. The other issue is that it can be more expensive to give an antibody than to give a vaccine. The antibodies delivered for oncology can cost thousands of dollars. Those delivered for global health can cost $100. We're aiming for ones in my consortium that would be $25 to $50 a dose. But that's still expensive, right? That's more than the income of some places in the world. You would gladly pay that to save your life in San Diego, but it might be beyond reach in some parts of the world. And we know that infectious disease anywhere will ultimately become infectious disease everywhere. And so we want to make sure that we're not just finding antibody therapies that are going to work here, no matter what they cost for the wealthiest nations, but ones that can also, and maybe they're different ones, and maybe they're the same ones, work in any part of the world to save lives and eradicate disease, to prevent infections from circulating anywhere around the world. In order to make an antibody treatment affordable, you need to do a couple of things. It's got to be really potent. You don't need so many milligrams to do the job. What you can make can be spread across more people. And if the molecule is easier to manufacture or grow in cell culture, you can make more for the same effort. So you can give it to more people. And we need it, if you're going to try to deliver antibody therapy to the far-reaching parts of the world, in addition to developed nations, we might need to think about molecules that are really stable, where if there's a breakdown in cold chain, we don't lose the therapy. Because we need to do two things. We not just need to treat ourselves, but we need to make sure that humanity overall is going to be cleared and protected from this virus. So to figure out which of these competing antibody therapies is best, which would serve our purposes of wanting to treat people in developed nations and save lives anywhere in the world, and prevent the virus from boomeranging around the planet every season, we need to find a way of getting scientists to be able to share samples and share data so we can compare the things side by side. The need for comparing them side by side is this, that if company A has discovered their therapy And they're going to go to the NIH and say, this needs to be tried in a clinical trial, and we think it's a good therapy because we've done experiment A. Well, company B is going to say, well, we have a therapy and we think it's the best, and we know because we've done experiment B. And company C is going to say, well, we have a therapy we think should be evaluated, and we know it's the best because we've done experiment C. The NIH is looking at this pile of data, and it might be apples to oranges. They want to evaluate things independently, and they want to evaluate them under the same assay conditions. So how's that going to happen? Well, early in February, the Gates Foundation called me and asked if I would set up a collaboration that would help them get this kind of data. And that collaboration is called CoPIC, the Coronavirus Immunotherapeutic Consortium. And it's housed in my lab in La Jolla Institute for Immunology. The goal of this collaboration is to find a framework by which all of these companies can analyze all of their different therapeutic candidates side by side under the same conditions to figure out which ones are best, what it is that makes them best, which experiments find the best ones faster, which ones are safer, can we engineer them to be manufactured more cheaply so we can treat more people, and not just treat Americans, but also save lives in other places of the world. So the reason they called me and asked me to do this is because I had done something very similar for Ebola virus a few years earlier. So when we started, and my lab had worked on Ebola virus and other emerging viruses until coronavirus, we had no vaccine, no drugs, and no treatments. Now, you're familiar with the shape of that virus. It's this long strand of spaghetti. And if you look at it in a higher resolution view, on the surface of that virus, just like on the surface of coronavirus, there are these molecules. So it's called spike on coronavirus. It's what makes the corona the crown. An Ebola virus, it's that glycoprotein there. They look like little mushrooms, little knobs, little trees stuck into the viral membrane, which is pink. Those are the molecules, whether they are coronavirus or Ebola virus, that the virus uses to attach to your cells and drive itself in. That is the piece of machinery that allows the virus to infect. Because it is what is on the surface of the virus, and it's what's exposed to your immune system, it's the target of your antibodies. Your antibodies are targeted to that surface protein called spike for coronavirus, called GP, glycoprotein for Ebola virus. And by targeting to that surface of the virus, they do a couple of things. By anchoring on there with really tight affinity, they might inactivate that piece of machinery. So if that antibody anchors on where the virus might otherwise bind its receptor, you've neutralized its ability to attach to a cell. If it anchors on in a place that prevents the virus from doing its unfolding spear fishing rod shape-changing thing it uses to drive itself into your membrane, that antibody is going to act like a club on your car and block any motion of that steering wheel. That antibody is going to prevent the virus from getting in. When it's bound on there, neutralizing or inactivating the ability of the virus to get into your cell, it's also using that constant region, that's the base of the Y, to signal to your immune system saying there's a virus here, come and kill it, and it clears the virus or clears infected cells from your body. So my laboratory is a molecular biology laboratory, and what we specialize in is called structural biology. We use X-rays and we use microscopes. We use microscopes that have an electron beam that's 300,000 electron volts and is 11 feet tall to directly image these previously submicroscopic particles to see the structures of the molecules that the virus uses to infect and the structures of the molecules that your immune system uses to fight it. What I'm showing you here is our structure of that surface spike protein of Ebola virus in blue and white. And in yellow is the antibody. Now these are just the arms of the Y. We tried to get high resolution, we just used that business end, the arm of the Y, to anchor onto it to figure out how this antibody from a person who survived Ebola virus infection in Africa could work. And we had looked at structures to understand how these antibodies might neutralize or inactivate Ebola virus and all of its cousins, Bundibugyo Ebola virus, Sudan Ebola virus, Marburg Ebola virus. And we had a lot of good information about what parts of these viruses would be targeted by antibodies and how tightly they could anchor on there and which one of them would neutralize the virus best in test tubes in cell culture. But that wasn't enough. We're still missing some information. We had, as a research field in 2013, antibody therapies that could neutralize the virus beautifully in test tubes, but didn't protect living things. As an example of this one I've shown you here, where the human antibody is orange, is anchored onto that white surface spike protein. And we had different antibodies that would protect living things, but didn't register as neutralizing in the test tubes. And that was a mixture or a cocktail of these 3 blue and purple antibodies. And that was real surprising. The things that looked great in test tubes didn't save living things. And the things that would save living things didn't register as interesting in the test tubes. And so we thought, what are we going to do? There's a couple of pieces of information here. One is that our neutralization assays that we thought were so predictive apparently aren't. And the other thing is that what worked was a cocktail, 3 antibodies put together, so maybe that's important. Maybe we need to hit it from different places. Another piece of information was this. It depended on how those antibodies were produced. If they were produced in one cell type we call CHO, Chinese hamster ovary, you could deliver a big dose and save half the animals. But if you produce those antibodies in plant cells, Nicotiana, you could give a smaller dose and save all of the animals. And the difference between the CHO cells and Nicotiana cells has to do with the identity of a carbohydrate molecule that's anchored onto the base of the Y, the base of the antibody. And what type of carbohydrate that is influences the ability of the base of the Y to recruit immune system help. And so maybe something about that was why the blue and purple therapy worked and the orange one didn't. Or maybe we needed a cocktail, or maybe we weren't running the right kinds of test tube experiments to figure out what would work. So typically what my research field had always done was one lab working by themselves, or maybe one lab and 1 or 2 others, would make a panel of antibodies. They discover them from a survivor of infection. They would winnow down the ones they were interested in by doing cell culture neutralization. Which of these antibodies would block the virus from infecting in cells in a test tube? The ones that did that would get tried in mice. If they worked in mice, they went into guinea pigs. They worked in guinea pigs, went into non-human primates. So every one of these steps is like a funnel. The idea being to get down to your 1, 2, or 3 hits at the end. And we did that because we had to, right? You're not going to try everything you come up with in animals, certainly not in primates. You have to narrow them down to the ones you think you're most successful in some way. And we did it because it worked. Hits came out the bottom. But it was clear that each one of these steps is a funnel, and some things fall through and other things do not. And we don't always understand the things that don't fall through because we stop looking at them. And so there might have been something that would have been protective in animal models that it never got tried because it didn't fall through that first neutralization funnel. Maybe there's something that would have protected primates, but we never had the opportunity to look at it because it didn't protect the mice. And maybe that's not the right experiment. I mean, who cares if we protect mice from Ebola virus? We want to protect humans, things that look like humans. And so our first criteria of selection had always been neutralization, because we thought it could forecast what would work in living things in vivo. But it was clear that it wasn't. And it occurred to me that what fell through all the funnels was what they had in common. That's the mechanical inactivation part of the antibody. Whether when the antibody molecule bound onto the virus, it physically blocked that virus machine. It became like the club in the car. It prevented the virus from attaching your cell. It prevented the virus from getting into the cell. If that antibody did something that had to do with the biochemistry and the biophysics of that machine, that would work just the same, whether it was in a test tube or a rodent or a human, because that's just mechanics. What we had never appreciated was the ability of that antibody, the base of the Y, to signal immune system help, because the human immune molecules are different from non-human primates, which are different from rodents, and they were never there in some of the test tube assays of cell culture function. And so that had been a black box for us. It's something that my research field really had to study. And in looking at the set of molecules that we had to fight Ebola virus at the time, it was clear that we weren't quite sure what the right experiments to do were. We really needed to understand how to make an antibody therapy against this virus, because we thought the big outbreak would come. It did shortly thereafter. We needed to understand what would lead to protection, and we needed a statistically significant number of antibodies to prove that we got it right. Maybe we'd only looked at the outliers and not the rule. And so we started the study. We had things that worked in test tubes, but not in living things. Things that protected living things didn't work in the test tubes. We were missing information. We needed better experiments. We needed more samples. And the trouble was that everybody was working by themselves in a silo. And there was no way we were going to get enough samples to really understand what was happening unless we found a way for everyone to be able to work together. At that point, the large Ebola virus outbreak had begun, and it was infecting larger urban areas than we had been seen before. And there was an escalating pandemic. And so we needed to do a couple of things. We needed to be smarter than we had been before. We needed to find a way of working faster, and we needed to find a way of working together. Now, it makes a lot of sense to do those three things, to be smarter and to work faster and work together. But the issue was doing all three at the same time. Because the time it takes to organize into a group and work together can slow you down. We know that competition is what drives science faster. Competition drives innovation. Competition, the not wanting to be scooped, the wanting to get the answer first, gets you the results faster. And we definitely need faster. We also need smarter, right? And then we all know situations in which working as a group stifles individual opinion and can lead to groupthink. And so we needed innovation, we needed individual opinion, we needed new ideas, but we needed to find a way in which we could get enough samples and enough tools to figure out what we were missing. What we needed was to find a way of doing all those things, three things at the same time. We needed to find a way of collaborating that could still celebrate the speed and innovation of competition. We needed to find a way in which collaboration and competition could coexist in order to take advantage of the best of both. And to do that, we needed to find a way to get a broader set of samples, a greater set of expertise. We're clearly missing information and not doing all of the experiments we needed to do. We needed to find a way of comparing everyone's therapeutic candidates side by side. We needed to do all of this a whole lot faster than we had ever done before. And so we devised a study by which we blinded all the samples. So to protect investigators' ability to publish, you've heard of publish or perish, and to protect companies' investments in their molecules, we would give everything a code name. So nobody would know whose antibodies were whose, and everything would be very fair. The samples would come into my lab, they'd all be code named. And then we'd pack identical boxes of the world's samples and send them out to anybody who had a different expertise and asking a different kind of question about a way that an antibody might work or a way you might test it. And so everybody did whatever kind of experiment they were an expert at on the same set of antibodies. They did them simultaneously. We were able to combine all of that data together into the broadest, deepest data set on Ebola virus that had yet been compiled. And with the understanding that we needed to move really fast, we did two things. We had a rapid study track, which was a proof of concept, and initial molecules that looked good, we would mix and match and see if that would find a better therapy. And we had a comprehensive track where we would take all of the antibodies and all of the experiments and run them all side by side and see if we could figure out a better therapy and a broader set of information. So with that idea of blinding the samples, bringing in more experts, more minds, a multidisciplinary study, protecting intellectual property, we launched this global collaboration called the VIC, the Viral Immunotherapeutic Consortium, also headquartered in La Jolla. We combined the expertise and samples of 44 labs across 5 continents that were academic, government, and industry groups into a single effort to figure out what antibodies against Ebola virus are best and how do we find them faster next time. And so over a couple of years, we generated this data set where on the vertical axis are the antibodies we gathered, and on the horizontal axis is every single thing we measured about those. We were able to make network correlation models to say of all the different features of the antibodies, which ones track together. If it has a certain kind of carbohydrate on that base of the Y, does it do certain things? Is that good? Is that bad? If it binds a certain place, does it have a higher affinity? Does it link to the ability of that carbohydrate to function? And we could come up with a logistic regression model that explained which things were most important and whether or not an antibody would deliver protection. As we could calculate protection as the equation on the bottom. P equals 1 over 1 plus E to the whole bunch of terms. And every one of those terms has a beta and a chi. And the chi is a certain feature or thing we tested about that antibody, and the beta is the importance of that feature. And we found of 17 different experiments, we could run high throughput and test tubes, super cheap, super fast, and cells, put that answer together, and you can predict what's going to protect a living thing with 96% accuracy. So area under the curve, 0.958. And so I'm showing you a bunch of bars here, and there's red bars and there's blue bars. The blue bars are good features about the antibody. Those are, if the antibody does those things, we think it's going to protect. The red are bad features. If the antibody has that feature, it is a bad thing. If it's a long bar, it's a strongly predictive feature. If it's a small bar, it's not that important. So what do we find? Well, what the field used to look at, that first step in the funnel, was where we have previous focus. That was whether or not it would neutralize a certain model in the middle, where it bound, and how tightly it bound. You can see that's not the most predictive thing. Instead, what we found were new ways of measuring the mechanical activities of whether that antibody could be the club in the car and anchor that machinery together. And a whole bunch of other things that we hadn't previously had good ways of measuring. And those are all the ways to recruit the immune system to come and clear the virus and clear infected cells from the body. The ability of the FC, that's the base of the antibody, to function. That was what that data was telling us about whether it was made in a certain cell type and how big a dose you needed to deliver protection. So by putting all these people together to do a single large study properly, we were able to write 78 papers and confer 10 PhDs in the knowledge gained by the study in the 3 years. We were able to revise the research pipeline to do better experiments, focus on new things. And we came up with information that yielded 6 new therapies. You probably read in the New York Times that suddenly there were treatments for Ebola virus when there wasn't anything before. This is one of them. This is the top performing cocktail. And this is a molecular structure from my laboratory through one of those 11-foot tall microscopes. You're looking at each one of those gray arms sticking out as a topographical map of the pattern of electrons that show the molecular structure of 3 different human antibodies. So it's a 3-part glycoprotein spike, and so 3 times 3 is 9. You have 9 copies of those human antibodies on there. And we can look at what each one is and where it binds and what its contexts are and understand how each one of those works. So by understanding what the right therapy can be, you can find them faster. And so that was the study I was in the middle of running when this broke out in winter. And in early February, it's amazing, it was only 6 months ago, the Gates Foundation called and asked if I would set up that same study for them to understand how we can find antibody therapies against the coronavirus faster. And so the initial funding from this is from the Therapeutics Accelerator of the Bill and Melinda Gates Foundation. They also had some input from Wellcome and MasterCard, and even Madonna put in some money. And the goal of this consortium is to gather the antibody therapies, just like we did for Ebola, and evaluate them side by side, figure out which ones are best, which ones we think should go into humans, and to engineer them and try to make them even better. So the samples come in, they're all given code names, they're all blinded, and that protects their intellectual property and it protects their investment. But they get a benefit. All the data we gather in this whole study, the companies can have back, and they can use that for their investigational new drug filing with the FDA. So there's a benefit to the company. By contributing their samples, it gets them that independent evaluation they need to be looked at for some of these clinical trials. And then whatever data we generate, they can have back and they can use it. Because we want these therapies to be used. We want these therapies 3 months ago. We will, at Jolla Institute for Immunology, I'll go out and distribute these antibodies and then analyze them in the same way we did before in lots of different kinds of strategies using lots of different brains. We're going to catalog all this information in a database called CoPIC. And this is run by my colleague Björn Peters at Jolla Institute for Immunology, where all the data we generate is going to be available real time to researchers and clinicians and stakeholders to try to understand what these antibodies are doing and how they work. And because the antibodies are codenamed and blinded, we can make this data available immediately. And we're going to analyze them, we're going to try to improve them, and we're going to recommend 1 or more therapies that we think should be used. So we have a couple of goals. The primary goal of the foundation here is to make 1 or more antibody products that can use to save human lives. We are looking mostly at cocktails of 2 different fully human, so a molecule just like you'd make yourself, neutralizing antibodies that are against SARS-CoV-2 to prevent COVID-19 disease. And we want to optimize these antibodies to make sure that they're durable and they're going to last a long time and we can deliver them broadly. Now, because I am an academic professor, in fact, you can see my professor robes in the background here, I like information and data. And I think that while we've gone to the trouble of convincing 50 to 70 different companies to give their samples, we have an opportunity here, and we need to make hay while the sun shines. We have an opportunity to generate a lot of data about how different kinds of antibodies work against this virus. We're going to be able to make in that database the whole landscape of what antibodies bind to what sites and how tightly they bind and what they do when they get there, and which kinds of antibodies are complementary to each other. So if we want an additive or synergistic cocktail, we can find it. We could also then use this data to evaluate the vaccines and figure out, when the vaccines are tested, do they make antibodies like these or not like these? What do they make? And then we can use these antibodies as discovery tools for more research and figure out how we can have antibody therapies that are not just good this year, but are good for next year and the year after if this virus continues seasonal outbreaks, or if, God forbid, there's SARS-CoV-3. So our use cases are these. The primary use case is in treating mild disease to prevent it from becoming severe disease. And so early treatment's better. It's like antivenom. We want to find individuals that are early in their infection that have a likelihood of progressing to severe disease because of their age or their underlying condition. But with enough doses, we can also use this therapy to protect healthcare workers and frontline workers, especially if there isn't yet a vaccine, to protect high-risk groups like pregnant women and others, and to circle up disease outbreaks. If the virus has reemerged among cluster of people, can we treat their contacts to prevent spread from that site? And so you might be asking, is it going to work? And it is. And after a couple of months of a lot of phone calls and a lot of talking and a lot of documents, we're getting the samples rolling in and these assays are rolling. So we're going to start generating all of that data to build that database. But what is happening in the world is that this virus is still on the rise, and we are not safe until we are immune. In order to be immune, we need antibodies, we need vaccines, we need treatments. They have to be discovered rapidly, they have to be de-risked to make sure they are very safe, and we have to evaluate them to see if they're going to work. We are here. It's going to keep climbing. And so how are we going to get out the other end? In order to emerge from our houses and continue with work and school and human endeavor and everything else that we wanted to do this year, we are relying on immune defenses. We are either relying on our own immune systems to recover from the disease, or we are relying on our immune system to respond adequately to a vaccine, or we are relying on the immune system of another survivor to have made antibody therapies that can be used to save other people. And so it is human immunology that is the reason that humanity is going to survive this outbreak and everything others. Human immunology covers a couple of areas: how we fight off infectious disease, how we fight off cancer, how we do not overactivate our immune systems to not have excessive inflammation, autoimmunity. And studying these three different areas of immunology gives us complementary information. So for example, an antibody discovered in basic studies of inflammation that targets one of your molecules that turns up your immune response, IL-6, that therapy is used into treating the very severe overactive acute respiratory disease that's the more severe COVID-19. Cancers can use integrins to infect, and in a process of studying cancers, we've developed anti-integrin therapy. Maybe that'll be helpful in blocking the coronavirus life cycle. And in the same sense, viral infections and bacterial infections trigger cancers. You know, we're very clear on the link between human papillomavirus and cervical cancer. There are Epstein-Barr virus can cause certain kinds of lymphoma. We're also certain viral triggers of autoimmune disease and inflammatory disease. So by understanding different parts of the immune system, these viral probes teach us how our cells work and have cancer. And viral probes can teach us, by following what the virus has chosen to target or erase or silence, we can learn how to control inflammation to prevent all the other diseases that we still get every year. These are my colleagues at the Institute, roughly placed according to our primary focus of study. And the very nice thing about having all these sources of expertise in the business, in the building, is the opportunity for the different areas of focus to illuminate each other and to find ways of going forward, not just coronavirus this year, but diabetes and cancer and heart disease and everything else that has to do with immunology and inflammation. So I will stop sharing my screen and I will now look at your questions.
Yes, we have a couple of them. I'm excited.
Okay. We know COVID-19 enters through nose, mouth, and eyes, can enter through broken skin or sex. I don't see why it couldn't. Viruses are crafty, they get in lots of ways. How likely is it the virus will mutate by the time vaccines become available? It already has. A mutation that arose in March has become globally dominant now in the world. It is a mutation that causes the virus to be fitter and grow more robustly. It does not make the virus more lethal or cause more serious disease. So we dodged a bullet with that one, but we want to keep an eye out to make sure another mutation doesn't happen on top of that one. So we have a fitter, stronger virus that does cause worse disease. So it already has mutated. It will continue to mutate. And most mutations don't do anything, but every once in a while there's one there is. Well, multiple vaccines need to be developed since they seem to be multiple strains of COVID-19 with different symptoms. I would say, yes, we need multiple vaccines, but that's more because we don't yet know what strategy is going to work. And not all types of vaccines will work in all people. If you're immunocompromised or if you're a baby, different kinds might not work. Like you might not be able to accept an attenuated replicating virus. The different symptoms has to do more with your individual immunology. Your microbiome can influence how you responded to certain infections, what you've been infected with before. Maybe you've recently had some other common cold coronaviruses, and that gives you some weaker cross-reactive T-cell response that could help. Is there an assay for neutralizing antibodies against the COVID-19 virus? In the laboratory, there's lots of assays. We can put antibodies into cell culture and see whether or not they neutralize. There are, I don't know if those are available commercially. They might be. There is a pseudo neutralization assay. When I say pseudo, what it means is it uses the receptor binding domain of the spike. So it's the bit of the spike that it would use to anchor onto your cells. And antibodies that anchor into that place typically neutralize because they're very good at blocking viral infection. And so by doing a kind of an ELISA sort of test or a lateral flow one that looks like a pregnancy test, antibodies against the receptor binding domain tend to correlate with having neutralizing antibodies. And there are lots of tests like that available. What systems or animal models did you use to determine that the antibodies worked for living things but did not neutralize in the test tube? The gold standard is non-human primates. And an open question in the field is, we need to find ways of using fewer animals and better understanding the output from these animal studies. What are we really learning when we evaluate something in a mouse? And can we make better models so we get better kinds of information? Fantastic presentation. Hey, thanks. As a leader in your consortium, how do you help competitive and driven researchers to collaborate for the greater good? Oh my God, I spent so many years thinking about this, especially with things like clinical trial embedded or premium. I have spent years thinking about this. I have a TED talk on this subject. Because here's the thing, to be really, of course everybody should collaborate. Of course everybody should contribute to the greater good. But you know, you got to be practical and know that even if the scientist had wanted to, the lawyers of that company might not let him, or the lawyers of his university may not let him, because they have invested a lot of money and there's intellectual property. And so you have to be really careful at protecting all those things, and you have to figure out what it is they're worried about. Are they worried that their sequence or their information will get out? Well, then you have to just make sure that they don't, that you were in possession of no secret information. You codename the studies. Are they worried that you're going to write a report that says that their therapy isn't any good? Well, by being blinded, I can write no such report. The report I'm going to write are that antibodies that bind these places and have these characteristics are good. Antibodies that do these things don't look so good. It's just a report of data and information. I will not unblind the study unless the owner of the antibody tells me I can. Now, the people running the clinical trials will need some of this information because they need to be able to make good judgments about what will be tried in people, and so they will be unblinded. but because I am the microphone and I am the paper writer, I am only looking at data. You have to find out what it is they're worried about, and you have to find a way of addressing that. And there has to be a benefit to them. So the benefit to them could be that one thing is that this study could be the gate they need to get through to get that independent evaluation of their data. It might not be enough for them to just say, we think it's great. You might need an independent lab to say, yes, it is, or no, it's not. We have so many scientists working on this that they have done different kinds of experiments or experiments in different kinds of cells. That's additional data the company didn't have. And they might need some of that data for their FDA filing. Like they might need molecular structures, but not have the ability to solve them themselves. So by helping us, by contributing their molecules, they get something back. And we want to help them. We want companies that invest in global health to thrive because they want them to be able to stay in business, to come up with something against the next therapy. And our job is to pick among what they want. And what the Gates Foundation is particularly interested in is not just making sure that the richest people in America can get therapy. We're pretty sure that's going to happen anyway, but that impoverished people can be protected as well, because we are all vulnerable if any of us are vulnerable. And so they're trying to make sure there are molecules that are also going to be inexpensive enough that they can be broadly delivered. For those who believe they have had COVID-19, what is the optimal time for getting a blood test to check for antibodies? It's hard to say. There's a lot of individual variability. A few days after you will have IgM, and more days after that you have IgG, and IgM are low affinity and IgG are higher affinity. I would say, and you might not register a strong antibody response soon, and you might get a higher one later, but it seems like maybe they decline after a couple months, so I'd say 2 to 4 weeks. I'm not sure. Can the virus enter through the eyes? Yeah, I think so. I think a lot of viruses go in. Any portal, eyes, nose, mouth, is a portal for a small droplet to attach to a mucosal surface. Could coronavirus be spread through blood transfusions? Sure. If you had blood that contained coronavirus in it, you would contract coronavirus. And so I hope that they are testing blood donations for that. Will Fleet make a recording of this event available online? I don't know.
That's a question I can answer. Yay! Yes, we will. So in about a week or so, we'll have this talk on the Fleet Science Center YouTube channel. So be sure to subscribe to that channel so that you get notifications as soon as new talks are published.
And then Sarah wrote, thank you so much for the amazing and eye-opening presentation. Thank you. Anonymous attendee, if a company university refuses to provide you with their prototype, does that make you or the consortium suspicious of its efficacy or efficiency, maybe giving the astronomical stakes the world has right now? Right now, I don't have time to be suspicious. I'm just trying to gather all of the moving parts and competing people and get the data I can and the people that submitted their samples. I would imagine, though, that if you were the NIH or the WHO or, an organization tasked with figuring out what you are going to put into people and what you're going to invest money in, that you would want some information about them. Now, many of these companies have amassed reams and reams of data on their own. And much of it has already been generated by independent groups. So they sent it out to academic labs to evaluate neutralization in the BSL-3 with authentic virus. And they sent it to another group to evaluate it in different animal models. So they already have a lot of independent data. And I don't know all of their reasons. What can we do to support your important work? Oh, thanks for asking. I work entirely on grants, and most grants are not funded, and I spend a lot of time trying to write grants. And if I have the money in hand, I can go do research, and I don't need to write a grant. Anyone that's able to make a gift to support research really helps. So when that mutation emerged and became dominant in the world, I thought, oh my God, we have to make that spike, and we have to figure out if these antibodies are responsive against it. We have to figure out what effect that mutation has. And it was a private gift of a couple $1,000s that let me make that and start those studies. So private gifts are definitely an investment in making sure that we can all get out of our houses and go back to work, and our businesses can operate again, and our kids can go back to school. So if anybody wants to be involved in the work and invest in it and be a part of the solution here of getting humanity back to what we meant to do. You can go to either my consortium website, which is CoVIC, CoVIC.lji.org, or just La Jolla Institute's site, lji.org. And you can click and get some more information or meet a scientist or make a gift.
Okay, we have somebody who raised hands. I don't know if maybe the typing in the question doesn't work, so I'll see if we can get Vicky to say something. All right, Vicky, you're on. Did you have a question?
You're on mute, Vicky.
Oh, that's right. Hold on. I'll ask her to unmute. No, we're not hearing her yet. Maybe not a question after all. All right, I'll lower Vicky's hand and disable the microphone. Sorry, Vicky. If you can type your question and if you have a comment, please do so. Quick question, you mentioned a TED Talk that you have. Is that, just if we Google you, we'll find it online. I'm pretty sure people would like to follow up on that.
It's on YouTube. Let me see if I can put a link in the chat.
And while
you can hear me typing.
Yes.
Here is the link.
Perfect.
Oh, wait, it's going to start. If I put it in the chat, will you see it?
Yeah, if you click on all panelists and attendees, then we will be able, all of us will be able.
I've got it under all panelists. Ah, all panelists and attendees. There we go.
Exactly.
All right, there's the link. And I'm putting also the consortium website and LGI's website. It's a real nice institute, and there's a lot going on here for cancer, heart disease, autoimmune disease, infectious disease, therapies, vaccines, and understanding how it all works together. The human immune system is the most remarkably precise and complex system. We are only beginning to understand its power. And if we can do more research to understand how we heal ourselves, we can emerge from the yoke of disease.
Oh, that will be great. Okay, we are just coming up on time, so this is perfect timing. Erica, thank you so much for this amazing talk. I've seen you speak before and I was super excited that you would be joining us because I just think you do a fantastic job of explaining what you do, how important the work is, and how it all works. So thank you so very much for joining us.
Thank you for your time.
I do hope that our attendees will visit the website for CoPIC and for the La Jolla Institute for Immunology. They do some amazing work. Of course, Jeff, you just heard with COVID, but also with many other diseases. So they're definitely worth checking out. I would also like to encourage you to check out the Fleet Science Center website. We have many more talks scheduled, as you might have seen in the beginning presentation. We have one more COVID talk coming up next week, Monday, where we're going to talk a little bit more about testing and why it is important to deploy testing, especially for asymptomatic patients. So we'll have UCSD and the Rady Children's Hospital joining us for that. If you like what the Fleet does, we are in a similar situation as the La Jolla Institute. We also need support to get this work done. We have been closed for about 3 months. That has made it very difficult for us to keep all the programming going. So I put a little link in the chat for everybody to see if you are so inclined and have the means to do so to support the Fleet or the La Jolla Institute, or both of us. That would be fantastic. We appreciate your support, and we hope that you will join us for many more talks and information. The La Jolla Institute for Immunology also hosts talks on a regular basis, so make sure you subscribe to their newsletter just as you subscribe to ours. And with that, I want to thank you all. Erica, again, thank you so very much for your time and expertise.
Thank you, and thank you for your attention at home.
Thank you. Thanks, everybody. We'll see you soon. Bye.